Welcome to Bioratna. Dear students, I am Chandana Sengupta and for today's video lecture, we are going to discuss the topic post-transcription process in eukaryotes. Here you can see in this diagram, the diagram A, this is the diagram of transcription. Here you can see that this mRNA which is formed, this RNA, it is not called mRNA, rather it is called primary transcript. It is called primary transcript, or it is called heterogeneous nuclear RNA. It is called heterogeneous nuclear RNA. This heterogeneous nuclear RNA, it is not capable to start the translation. Whereas in case of prokaryotes, the mRNA which is formed, it can immediately start translation, but this heterogeneous RNA, it cannot do it because it has two types of sequences. Here this part, intron, it is the non-coding sequence. The introns are the non-coding sequence, whereas these exons, they are the coding sequence. So these introns, they are interrupting. Introns, they are interrupting the coding sequence. Coding sequence, they come in between coding sequence, and so this heterogeneous nuclear RNA is not able to perform the translation process. So the thing which is required, it is to remove these introns. To remove these introns, so it from this heterogeneous, it changes into homogeneous, containing only the exons and not the introns. This is one step of post-transcription process, that is splicing. So what is splicing? Splicing is the removal of this interfering introns, these introns which are interrupting the two exons. So removal of these introns and then joining together of the exons is called splicing. This is one. Other than that, there are two other processes also called capping and tailing. Please note one thing, the unsaid things. NCERT आपको बहुत कुछ कहता है. You need to observe that. What if the question is asked like that? See, when this transcription process means this mRNA is not yet released from the DNA, at that time only the capping has occurred. What is capping? Capping is the addition of, here you can see G, guanine. This is methyl guanosine triphosphate. So the addition of this methyl guanosine triphosphate to which end? This is also important. Always mention the polarity. To the 5' end is called capping. This is called capping, and this occurs even when the mRNA, rather we call it heterogeneous RNA, is not released during transcription. So what is the function of this capping? This is the 5' end. The 5' end, you know that there are nucleases, the enzymes nucleases which degrade the nucleic acid. So there is one type of nuclease called exonuclease. This is exonuclease. What is the function of exonuclease? These exonucleases, what they do? They cut the nucleic acid from the sides, not from the center, from the side. This methyl guanosine triphosphate, it protects this heterogeneous RNA from the action of the exonuclease. This is its one function. Its another function is, now it is eukaryotes. So in eukaryotes, the primary transcript, it is present inside the nucleus. न्यूक्लियस से ये बाहर निकलेगा साइटोसोल में, then the process of translation will take place. So this capping, this methyl guanosine triphosphate addition to the 5' end, it helps the mRNA to move out of the nuclear membrane through the pores. When it moves out, then it is mRNA. Fully वो post transcription process is completed, then only it moves out of the nucleus. So we cannot use the word hnRNA or primary transcript when it is moving out of the nucleus. So this was capping. So in this we complete this first diagram, this diagram A, where the heterogeneous RNA is capped. 5' end is considered as the head, and here the capping has been done. Now come to this diagram B. Here you can see that the heterogeneous RNA has come out. The next two steps means adenylation and splicing is not shown here. It has come out and it has the capping because capping was already done. So here the capping is there and it is showing you the exons and introns. Exon, intron, exon, intron, exon, like that. This is your diagram B, where the whole heterogeneous RNA is shown together with the capping process which is already done. Now come to diagram C, which is the tailing or adenylation. This is adenylation. What happens in adenylation? In case of adenylation, which is called poly A tail, about 200 to 250 adenine residues are added to the 3' end. At this 3' end, 200 to 300 adenine residues are added. This is called adenylation, and this adenylation, it helps, like in case of capping. Capping, it protected the 5' end from exonuclease activity. So adenylation process, it will help the 3' end not to degrade by the exonuclease activity. In this diagram itself, you can see that these introns, they have formed the loops. This is the starting of the process called splicing, where now these introns will be removed. So these introns, they have formed the loops here and the adenylation process has been completed. Now we come to the final diagram. The capping has been already done, so capping is there. Adenylation has already been done, so these are the 200 to 300 adenine residues. So in your final diagram, the post-transcription process has been completed. You can see the capping, you can see the tailing. This part which I have drawn in dotted line, this shows the different many adenine residues. So this is tailing, and here you do not see any introns. That means they have been already spliced off. And now this is called mRNA. It has changed from heterogeneous nuclear RNA into mRNA, which is now ready to do the translation, and it will be now transported out of the nucleus. So students, why you may ask that why these introns, they exist there when they do not have any role, and they are to be, मतलब ये झंझट क्यों कि why they are there and to be spliced off this process? This shows an ancient feature. This shows the antiquity, like the vestigial organs in our body. They were there, now they are not functioning. These introns were active. This is an ancient feature. Now they are not working, and before the work is done, they are to be removed. So these introns, they are ancient features. They show the antiquity. And this splicing, it shows the dominance of RNA world. It shows the dominance of RNA world. So this was one more topic from the very interesting chapter, that is genetics. Hope you have understood it properly. Then do subscribe, like, and don't forget to share it with your friends. Thank you for watching the video.